No.
You can get started. We are recording already and transmitting to YouTube. Many people watch through the YouTube. Go for it, Savio.
Let's start?
Yep.
Okay. Uh, Good afternoon, everyone. It's a pleasure to meet, introduce Dr. Gustavo Caetano-Anollés. He's a very good friend. In my opinion, he's one of the most important research in molecular evolution. Gustavo's current research integrated structural biology, genomics, and molecular evolution with an interest in evolution of macromolecular structure. His group has recently reconstructed the history of the protein world and suggests the existence of a big bang of protein domain combinations late in evolution. It's a very interesting work. And today, Gustavo will talk a little bit about the recent results of his research. I'm sure it will be an excellent lecture, and I hope that you enjoy. And Gustavo, thank you very much for staying here with us.
Thank you very much, Savio, for your kind introduction. Good afternoon, Brazil, and it's a real pleasure to have the opportunity to present the results of the research that we've been conducting over the past, I would say, 2 decades already. I'll talk today about the compressed molecular vocabulary of life, and it's a puzzling title, but actually what I'll be describing is how this vocabulary, that I'm going to be proposing exists in the molecules of life, can be dissected with deep time chronologies and networks. So you have a punchline there in the bottom of basically what the talk is going to be about. As Savio was introducing, and let's see if this works. Yeah, there it is. We've been exploring in my lab for more than 20 years the structure of molecules as a medium to access the history of those molecules. That have been the actual background, of the kind of research we do. It's all about a back-in-time exploration, that is idiographic, historical, retrospective, and that uses three-dimensional structure in all its inceptions as a window into function and evolution. So I'm really interested in function ultimately because molecules are just the mediators of the functionality of organisms, in all their complexity. So before starting, I want to dissect the title of my talk, isn't it? So in order to dissect it, let's define words. Let's define vocabulary. Let's define compression. What do I mean by deep time? What are chronologies? What are networks? So that's where we're going to be starting. So what is communication in biology? So obviously communication is part of the entire repertoire of the molecular makeup of cells and organisms and even viruses. And communication is the use of messages to convey meaning. We know very well about this because we live in the era of the internet, and communication is crucial, isn't it? So, and meaning, we need to convey meaning. For that, we need a language, a formal symbolic system of complex communication that uses signs and rules to communicate that meaning. And this vocabulary is part of that system, isn't it? It's the set of all signs used to communicate that meaning. And this is usually called the lexicon, isn't it? So it's lexicon what provides the elements for that communication to occur. And let's also define what is a sign, which is this semiotic entity that communicates meaning and is causally related to an object through interpretation. And we usually understand a sign as a word, for instance, isn't it? The word, as soon as you hear it, you understand in your mind what it means. So my goal today is basically to make analogies and use data from phylogenomics to understand this vocabulary, this communication in biology. So what is compression? You know, this other word that I'm using. Well, compression is a process of conveying the same message with a smaller number of signs. And so here we have the word hello, you know, so in different languages, hello, hello, hola, bonjour, hola. So probably the Portuguese version, hola, is the most compressed of the ones that you have there in that slide, isn't it? So, but we understand compression because as bioinformaticians, we know about that data compression. We know that sometimes in order to be able to transfer information or to work with information or to mine information, we need to somehow compress that information and distill something from it. And we know, for instance, very well about the lossy, glossy, and lossless compression that occurs with images. And we use, for instance, PNG versions because they're easily to embed in documents and they don't take much of space in those documents. So it's intuitive. As soon as you're talking about compression, then you start off thinking about data compression. But in language, it does occur, isn't it? We have contractions. We use apostrophes to replace letters. So we are compressing would have, for instance, would, or compressing by truncating words like captain. You know, this we are economizing by doing that. We use it also in expressions, in idioms, for instance. In Norman English, if I'm saying ignore a hand, you know. So in India, an idiom somehow also compresses information. So we understand that concept relatively well. We also understand when we go shopping, for instance, that we can find elements, objects of different sizes. But the object in itself is the same, isn't it? But just it is the size that changes with those objects. So that's something that is universal. You know, we see them everywhere. We also, some of you may also know about the Shannon-Weaver you know, exchange or hypothesis of how information is transferred, isn't it? So you have a sender, and then you have a receiver of a message, and what Shannon and Weaver proposed was that a way to transfer that information is basically to encode that information, usually compressing it, isn't it? And then using a channel of that communication, transfer it to the receiver end, and then using a decoding system, unfold that message again. And in that transfers, then we have to deal with noise on some way, and also on a way to somehow interact between the sender and receiver through feedback that would allow us for the communication to be stable and persistent. So that's an introduction of what is communication, what we understand by communication and by compression. So, because vocabulary is an evolutionary process, and here I want to stress some work from Mark Pagel in the UK. Words become dominant in evolution. This is something that has become a realization of languages. They arise mostly by frequency-dependent selection through models of diffusion of innovation, and they can be studied with phylogenies. As we study, you know, RNA or protein or genomic sequences. And usually when we build phylogenies like the one you see here in the left, these phylogenies are close to the ones that are ideal from a phylogenetic point of view. That means that little information is controversial in the building of the phylogeny when you are talking about languages. And that is not the case with biology. So that tells us that, that language evolve. They do evolve. The diagram in the left that comes from this paper from Pagel in 2019, where they are proposing the idea that, that we as a species use words disproportionately that other persons use. So that means that the words that are common to a large number of individuals, those are becoming the most used words that you have. And that allows us to propose that words like two, for instance, are really, or three or four numerical elements, cognates. These are very ancient. They've been widely spread at the beginning of our languages and spread widely. And they are quite distinct from others that spread through languages a more haphazardous way, like the ones you have here, bird or drink. Some of the roots of these words are common, but others are not. And they're not spread through all languages. So it means that they're more ephemeral, they're more recent. They've been recently added and are more of a decoration. So that's a central theme that already students have been kind of understanding. So what is a chronology in that title of mine? Well, it's time and logic, the logic of time, the science of arranging events in their order of occurrence in time. And here we have a word cloud that I've generated with Manfredo Sauferheld in my university, where we are basically using phylogenomic information to generate an ordered word cloud, where on the left it's very ancient times. There you have a 3.8 billion year mark, and as we are traveling towards the right, you are seeing approaching the present, isn't it? And there you have these words that have a meaning. They mean things for a biologist: cells, pyrophosphate, transfer RNA, genomes. And we are trying to find a time of origin for each of those and placing them into this conceptual framework of a word cloud. So my laboratory has been generating chronologies for a long time. There are chronologies of protein domains and loops. We'll be publishing one very soon, updated on protein loops. We are interested in chronologies of molecular functions, of metabolisms, of metalomes, of biocatalytic mechanisms. We are interested in other qualities, like, for instance, the speed of the folding of a molecule. We are interested in chronologies of RNA molecules, like transfer RNA, or the entire structure of a complex, like the ribosome that contains RNA and proteins. So we've been focusing on this for many, many years. The goal is basically to place that chronology, which is the history of biochemistry ultimately, into a framework that is global. And here comes James Hutton in the 1750s, which basically developed this concept of deep time, this philosophical view that changes are developing over the age of the Earth, isn't it? So life is a phenomenon of Earth as we know it. Now there may be life in the rest of the universe, but the life that we study is definitely Earth's life. And we need to place it in the context of what is called big history, isn't it? The history of the universe, the history of our solar system, the history of our planet, and the history of the conditions where life can emerge, which is believed to be between 3.8 billion years to the present. So I mentioned networks. So what is a network? It's a graph, a mathematical structure comprising a set of objects in which some pairs of them are related in some sense. So you're familiar with this as bioinformaticians. The networks are very, very popular these days. And why are they important? And important for me, at least, because we are searching for generative processes of biological complexity, and networks allow for us to explore that aspect of the equation. And to make an introduction to that, exactly that point of view, is I'm a genomicist, but I am also a systems biologist. And what does a system mean? This comes from Herbert Simon, Nobel Prize laureate of economy in the 1950s. If you, as biologists or bioinformaticians, have not read this paper that I'm listing there, you should. This is an incredible piece of conception, basically, that Simon put in 1960s. And there he defined basically what is a system. It's a structured. A system is a structured entity that describes this interrelationship of their parts. And obviously you see where I'm going to, isn't it? Because we are, after all, trying to understand the complexity of life. And in order to do that, we need a framework. And systems complexity theory provides that need, because we need to define those elements. So systems are, according to Herbert Simon, are organized entities that are structured in no simple ways. And here, from his work comes that famous phrase, the whole is more than the sum of the parts, that we are very, frequently hear. It's important from a pragmatic point of view, because it allows us to dissect the systems like biochemists have been doing. They break up cells into component parts and then they analyze the elements, and then they try to assemble all those elements that they've analyzed into and kind of interpret the whole, isn't it? So it's a of wholes, and that's why the title there, Systems, Parts, and Wholes, and also a way to understand how those parts are assembled into wholes. And here Herbert Simon also introduces the idea that systems are hierarchical. So what is a hierarchy? An organization that is ranked according to some authority. That's a typical definition. But let's read what he says in that paper: a system that is composed of interrelated subsystems, each of the latter being in turn hierarchic in structure until we reach some lowest level of elementary subsystem. So this is interesting because, of course, he was looking at a company level, but in his paper also he talks about proteins and DNA makeup at 1962. Please look at that time in the publication of that paper. Clearly, systems, if they're hierarchical, there are levels, there's nesting, there's balance in that hierarchy, and there are authorities, and all those need to be somehow dissected. And we have the tools, because in the 1950s also there was a revolution in mathematics in terms that network theory was developed, and a network has these two components: the nodes, the parts of that growing system, and the links that describe those interrelationship of the parts. And that allows us to basically use that mathematical framework to understand systems complexity and make it explicit. So that is an important aspect I wanted to stress. A second aspect that I wanted to stress before entering into the idea of language and biology is that the evolution is ultimately a process that describes how systems grow. And as soon as you start looking at the literature, you're going to see papers like this one published in 2014 that studies how the Tyrannosaurus family members have grown in evolution, you know, and they started with small dinosaurs, and then those dinosaurs increase in size. The same occurs with viruses. The evolving capsids have increased in size in the virus group, so there is a rationale for that growth in some way. There is also considerable knowledge supporting the idea that biodiversity is also evolving and increasing in diversity. So there is paleontological evidence supporting a pervasive increase in the number of species with time. So that means it's not that systems only grow, but complexity grows also in evolution. Even the ribosome has been evolving in time. So that means that at the molecular level, elements that are making that cellular complexity that are molecular are also increasing in time in size. You know, and when I'm saying increasing in time, I'm saying billions of years, of course. No, this is here we're talking 3.4 billion years of evolution. Now this that I call accretion, which is a process of growth and increase, typically by the accumulation of entities and their interconnections, it's not new. It has been there with astronomers for a long time, isn't it? That's how we modeled the origin and evolution of our planetary system, for instance, there. It also occurs in networks, isn't it? The process of growth and increase of nodes and links defining a network system, including their associated properties. And we know about this because we've seen the internet growth. Or the World Wide Web grow from small few connections to the enormous connectivity that exists today. So it's not an alien concept either, and we understand that this is occurring. As soon as a system is growing, as a network is growing too with it, isn't it? So it occurs in galaxies, in stars and planets, in molecules, particularly in cities. The fabric of a city is so cohesive that very few cities, once they're established, disappear. They can be replaced, like Mexico City, isn't it? You know, Tenochtitlan disappeared, but then a fervent city grows from it. But the city is a perduring object. Also very much as molecules, stars, and planets. So we've been looking at accretion at a planetary scale with a focus on molecules. And here I have a kind of intuitive slide, isn't it? So we have at the bottom, Earth changing with time, isn't it? The Hadean Earth, the snowball Earth, today's Earth. As Earth has been changing, life has been appearing and changing with that evolution, isn't it? So if we focus on cellular makeup, we understand that everything started with primordial cells in some sort, and that primordial cell at some sort reach a threshold level that we usually associate with the last universal common ancestor of our life, Luca, as is centered there in green. But then those cells kind of unify into multicellular entities, and those multicellular entities become more and more structured into higher organisms. The same we can say with molecular world, for instance, with proteins. First origin of proteins are primordial enzymes, simple enzymes, rather haphazardly made. And then these enzymes become more and more sophisticated with the entry of the genetic code, the entry of aerobic metabolism as soon as the snowball Earth generates oxygen in our planet. And then mature complexes like the non-ribosomal protein synthetases that generate all these incredible small incredible diversity of small peptides, you know, that are fundamentally necessary for life at multiple levels. And at the top there, it's our own work on the history of the ribosome, how it starts from small few helices that assemble to produce a machine that would allow us to synthesize proteins in a more effective way, and how that machine has grown in time till the present. So this is kind of a summary slide of a number of things I'll be presenting today. As soon as we are studying accretion at a molecular level, which is the focus of our research, then you need to choose the polymers of life. There are many, but definitely proteins and nucleic acids are the ones that are encoded in our genomes, and in a global way. Of course, genomes encode enzymes that are responsible for lipid biosynthesis and all that diversity that exists. But it is proteins and nucleic acids that are really the workhorses of the cells, and those are definitely interesting subjects for exploration, isn't it? But now, you can study this at different levels. Let's say if you're interested in proteins, like I'm kind of visualizing here, you can study the primary sequence of a protein on the left there, isn't it? So you're focusing at really lower levels and high speeds of change at a planetary scale. That's a calculation that I made many years ago. 10 to the minus 15 years in terms of change of a new amino acid, incorporated in the sequence of a protein in a planet, versus 5 times 10 to the 7 years for the makeup of a ATP synthase motor. So that means that look at the breadth of this from microseconds to billions of years. And in the same way as this phenomenon is occurring, that means that you can choose and pick where you want to study things, from a phylogenomic point of view. And we've been picking the domain level and the tertiary structure level as our best candidates to study the complexity of the world. And the reason is that it is at that level where you have these modules, these elements that appear in structure, that are structuring basically the molecular makeup of life. And these are successful architectural designs that are repeated over and over again, in different contexts. That means that you can have them in a bacterium, but you can have them also in humans, or you can have them in a plant. And these elements, some of them are, look at the supernary on the left, these are beta hairpins. This is a small repertoire of them exists, but they're repeated and arranged in different ways in different folds of the proteins, as you see them there, like for instance, a timbarel and a ferredoxin fold, they will have a beta-alpha beta element that will help make up their complex structures. In the same way, at the RNA level on the right now, you have the helical elements, the stems, as the major elements of folding of an RNA molecule, isn't it? And as soon as you have those elements, then these elements, which are modules, need to be arranged with other modules to build that complexity. And in doing so, they generate another kind of module, which is called the junction, which somehow arranges in 3 dimensions the individual helical elements. And then you add other motifs, A minor interaction, tetra loops, other elements that are solidifying the structure of an RNA molecule. And all of them are interesting instantiations of molecular structure that can be studied at a phylogenomic level. So how do we do that study at phylogenomic level in order for us to construct these evolutionary chronologies, as I was saying at the beginning? If we are to mine parts and wholes of systems, then we need to build phylogenetic trees of wholes and parts. Why phylogenetic trees? Because that is the standard way that we've used during the past many decades to understand evolution, isn't it? We should use networks, but unfortunately, computationally, we cannot do that. So we use specific instantiation of a network, which is a tree structure, to generate a map that tree structure in time. And so our goal is to build, given a genomic census, for instance, of some feature. It could be anything. It could be a transposon, or it could be a fold of a protein, or it could be a conserved motif. If we survey that and we build a phylogenetic data matrix from that, where we would have wholes and parts being represented there. Let's say I would want to build a tree of proteomes based on domain parts of the proteins that make up proteomes. So I will use domains and proteomes as the elements of the building of that data matrix. And that allows me to build trees of wholes and trees of parts. And that allows me also to use character state reconstruction, that CSR little abbreviation that I have there, to also study changes along the branches of those trees. And both strategies are useful. You can actually, with both of them, generate ages. And the ages can be used to visualize ages from a global perspective of the whole. So that means we could study the age of the individual components of the proteome by studying the ages of the domains, and then make a statement of evolution from that. In all cases, having ages allow you to use those times of origin to build chronologies. And then the chronologies allowed us to trace them onto the wholes. And these tracings of parts and wholes provide us incredible view of evolution that was not possible before. So here we have the two main strategies to study parts. So when you are studying proteomes, for instance, you can build a tree of proteomes. So A, B, C, D, E, F are proteomes. And the tree describes how domains, for instance, which are indicated by numbers there, are changing and defining the tree structure of the tree of proteomes, isn't it? And so we can locate individual changes in the branches of the tree, and by locating those changes in that rooted phylogeny, then we can infer a chronology. 3 comes first, 4 comes later, 5 and 6 come afterward. So this is a partial chronology. Why a partial? Because we can't access things that have been occurring below the root level of the tree. Now, we can solve this problem by building just a tree of domains. Instead of looking at trees of wholes, let's look at trees of parts, build the trees of parts from the phylogenetic data matrix, and there we have a full chronology that studies the origin and evolution of all the parts. From there, 1, 2, 3, 4, and 5, and 6 can all be defined directly from the actual taxa in the tree, because those taxa are the parts that are evolving in that tree. So that makes the big difference in building chronologies, and that's how we work out to build chronologies of proteins on the left, especially protein domains, and RNA parts and RNA molecules on the right, isn't it? So here we have wholes are proteomes in the left, characters are protein domains in proteomes. In the right, we have wholes are molecules. Let's say this is a 5S ribosomal RNA on the top, and the characters are molecular substructures in those molecules, isn't it? They could be helices, it could be elements of stability. We can use any kind of molecular character we want in order to build this kind of trees. And if we build trees of molecular substructures, then that allows us to build tracings of ages of time of origin of the substructures into the molecules, as the one you see on the bottom there. The same with proteins. You can study and trace the age of individual domains. Like, for instance, the domain that is in red there in the left is a domain of a protein that is very ancient. That's the catalytic domain of an aminosyltRNA synthetase. So it's the origin of that molecule, and we can do and dissect this with this kind of approach. So here is the output. This is the matrix, isn't it? Visualized as a heat diagram. The more reddish it is, the more abundant it is, and we use abundance as a character feature, a quantitative character. It's quite distinct from the the other characters that we are used to utilize, like just in sequence, for instance. These measure the abundance of an entity in a whole, a component in a whole. And so here we have rows of proteomes, columns of structural domains. And if we study taxa, the wholes, then we are going to be building, trees of proteomes. So a proteome will be the collective of all the proteins. Like, for instance, this is the collective of all the proteins of the SARS-CoV-2 proteome. They're responsible for COVID-19. So that's the whole. And we can study how that whole as a unit is evolving in time. And we've done this for many years already. Others have done the same thing. This is the group of in Bristol of Julian Go that has been generating automated phylogenies based on domains that provide a view. A tree of life. These are tree of lives, isn't it? There you have bacteria, archaea, eukarya. They're good approximations for the history of organisms, the trees of proteomes. Why? They're much better than ribosome because here we're taking an entire repertoire, isn't it? The entire set of domains of an organism. When you are studying the ribosome, you're studying the 16S component of an incredible complicated complex that has proteins and has other RNA molecules, isn't it? So it's really limiting. If you focus on a whole, then the whole provides a much more much more representative view of the history of an organism. So this is another one that we've generated. Here we are proposing to add viruses to the tree of life. And we find that viruses appear at the base of our trees of domains. This is a maximum parsimony tree that we've generated. And on the right, this is what we call the evolutionary PCA analysis. This is a numerical analysis of the ages of the component parts of a proteome. When you do this, now you have a three-dimensional view that expresses time in it. So you can see at the bottom there, that's the origin of life. Then viruses, the little star is last universal common ancestor of cellular organisms. And then you have bacteria, archaea, eukarya unfolding all the diversity with time. You can see that unfold in a better way. This is a phylogeny generated from domain structure. And on the left with a tree representation, and here at the right you have the evolutionary PCA analysis that shows how an origin below viruses, then generates the viral group, and then this viral group generates the diversity in bacteria and archaea in terms of proteomes, and then eukarya, and then you can see they're separated. Clouds of evolving proteomes and I'm saying evolving because they have different ages that locate them into this three-dimensional view. So remember, it's not the actual features that we're studying, but the ages of those features that we've been studying and we are unfolding in this diagram. So the interesting thing about this is that because of this arrangement in a tree of life and an evo-PCI, there is a last universal common ancestor of all life that includes viruses. And then we need to define another ancestor. We name it Lucella, and a last universal cellular ancestor, because now we have two elements that need to be accounted for, not only one. Luca, the last universal common ancestor concept was a concept for cells only, but once you add viruses into the mix, then you have to reevaluate everything and see, well, now we have two elements to understand in this makeup. Now, we can also study parts and generate trees of parts and this is a tree of parts from the Cath architecture classification. These are the trees of architectures. These are generic designs, and they are just a limited number, the ones that you see here in the chart that define all possible folds. And it turns out that there is a history of those folds, and this history somehow describes this tendency go from simple unordered structures, like the ones that you will see in sandwiches and bundles, all the way to highly complex order structures, like the propellers. The propellers are incredibly sophisticated architectural designs that exist in proteins, and those proteins are particularly located in eukaryotes. So it is a structural development that occurred in an advanced group of lineages on Earth. Well, of course, if you have a chronology, and if you want to turn that chronology into deep time, then you need a molecular clock, isn't it? Something that will place the chronology within a time frame of billions of years, and that allows us to then relate molecules to the makeup of Earth as the Earth has been evolving. And so here we have a tree of domains on the left, and that tree of domains turns out to be somehow expressing ages, ages of protein folds that are correlated with the time of origin of those molecules. And how do we find the time of origin? Well, we look for fossil evidence, molecular markers embedded in fossilized information, in crystals, in tars, and there's a lot of information that's coming that you can take to use and to propose that molecules are somehow linked and correlated to those elements. So this is how we've generated a molecular clock of folds, and the molecular clock of folds obviously provides a different time frame, a real time frame for the chronology of evolution here of domains. So the tree of domain now has a different context. Now we can map on top of it for instance, the great oxygenation event of Earth, appearing just in time when fold structures associated with domains that are not universal or not shared between viruses and cells, but specific to groups, like those of bacteria in green and light green, for instance, there will appear. So it makes a lot of sense. It is here where at some point the first organisms, diversified organisms, are appearing on Earth. And before that time, you had commonality. These are the structures that are universal. They may be lost in some lineages, but that's okay. That's not a problem. That's part of the noisy aspect of evolution, isn't it? So, and that allows us to put forth a chronologies, and chronologies of very ancient cells that gave rise to viruses and cells, the appearance of cells, appearance of a stem line that ultimately gave rise to individual lineages, and the first ones in bacteria, and then the rise of viruses, archaea, eukarya later on in evolution. So all this makes a lot of sense biologically, and you can derive it directly from the chronology of trees of domains just by looking at these taxonomical groups that we see. The groups of domains that are universal, that are only universal to cells, that are shared by individual superkingdoms like archaea bacteria or bacteria and eukarya, or that is specific to archaea, specific to eukarya, specific to viruses, and so on. So that's an interesting thing, and it also makes sense because if you, for instance, map one subclass of domains, those associated with multicellularity, you see how multicellularity, those parts of domains, domain parts of the proteomes, arise massively at a time when eukarya as a superkingdom is developing multicellular structures. And that's where you have fibronectin, invasins, intimins, and so on. These are just markers out of 400 structural designs that are linked to multicellularity. So this is rewarding. As soon as you see this, and as you see many other things that you can map here, it makes sense. You can see how evolution goes gradually enhancing innovations, adding elements, new elements of structure to its makeup, and not losing it. It may lose them in lineages, but globally, at a planetary level, it just accretes, adds more and more wonderful components to that makeup. Okay, so, well, we were concerned to understand why you will have these modules arising, So we need to understand why modules arise to begin with. So we proposed with Jay Mittenthal and my son many years ago a model, and the model is this biphasic or bow tie model, where biological structure is driven by a double tail of accretion and change. So first you have accretion, bring something that brings together disparate parts to form bigger wholes, and then you have a second component, change, providing opportunities for growth and innovation. And that's a small little diagram that describes the rationale. In a first phase, parts are at first weakly linked and associate variously. As parts diversify, they compete with each other and are selected for performance. The emergent interactions constrain their structure and organization. This causes parts to self-organize into modules with tight linkage. So this is kind of, this first phase is a phase of module generation, isn't it? But as soon as you have the modules, then the mutational process is responsible for change, isn't it? So the modules become variants, and the variance of the molecules, very much as the variance of the genomes of the COVID-19 virus, isn't it? So it's a process of change. And these modules then evolve and become new parts for a new generative cycle of higher level of organization. So that means that we have, let me go back. So we have a first phase and a second phase. A first phase of module generation. Modules are still universal at this phase. And then a second phase where things become diverse and change. And just by looking at the Venn diagrams of modules that we know exist, like protein loops, like domains in proteins, like gene ontology terms, like RNA families, like ribosomal proteins, enzymes, virus domains, virion types, and we have many, many more examples that I'm not showing here. In all cases, you see this red number there, sometimes quite massive, like in the case of protein loops. The more ancient the elements are, the more massive that number is, like in the case of protein loops. And that number is a universal descriptor, isn't it? It's the number of protein loops that is common to archaea, eukaryote, bacteria. So if you're thinking of the diversification life, you have a phase one where you generate this commonality, and you're generating the modules, during that phase as time progresses. And here you have that little diagram there on the left. And then you have a phase two, and then this phase two, then you have diversity appearing, and then this is where lineages of proteomes appear, and that's why protein loops all of a sudden are distributing in the different groups there. And as soon as you start looking at all these diagrams, you see incredible commonalities. Like, for instance, the bacteria and eukarya shared group, Venn group here, is always supernumerary compared to the rest of things shared by two domains or superkingdoms of life. And that's a commonality. We've used this commonality to support, from a comparative genomic point of view, the pattern of evolution that we've been seeing. So with Jay and Derek, my son, we've been, at that time, we dissected and tried to find support for the model. And well, we find support in development with the developmental hourglass. We found support in the diversification of macromolecules, protein structures, shapes of RNA molecules. There are theoretical studies that support this kind of proposal. Diversification of networks, early metabolism, evolution of codes, evolution of development, and epigenetics. In all of those cases, we found examples that would support the biphasic theory. But I think most of the support comes from the actual chronologies and the chronologies of networks that we've been generating. So this is a chronology of proteomes, and you can see the biphasic element, how domains are at first universally appearing at the beginning there, at roughly up to 2.5 billion years ago, and then it decreases and then increases again. So there's a biphasic element in the second phase, that includes epoch 2 and epoch 3. There you see diversity. There you see all the different domains apportioned into the different lineages of life appearing in evolution. So it matches our hypotheses. So, of course, the hypothesis then can be turned into a network hypothesis, isn't it? The double tail predicts the evolutionary emergence of network structure that is both hierarchical and modular. Why? Because at the beginning, you have a competitive optimization of parts, which are the nodes, and then the nodes assemble into groups, and those groups are the modules. This is the module emergence element, and then those modules then diversify in a novel way to generate new parts, and these new parts, higher levels of structure, are parts that are higher level in a hierarchy. And that's why you need to find hierarchy in order to support this. And that hierarchy needs to be modular. So you need to evaluate hierarchy and evaluate modularity in a network in order to test whether the biphasic theory is correct. So here again, it's put forth in a different way. During phase 1, the emergence of hierarchical modularity gives rise to small, highly connected subnetworks, which can then become modules. And then during phase 2, these modules coalesce by combination into higher modules of network structure, all of which enhance the cohesiveness and stability of the network structure. So this is a proposal that, that somehow follows directly from the biphasic theory that we've put forth in time. So we put it to the test with chronologies of networks. So given parts and wholes, we can generate networks, and we can trace those networks through events. An event 1, event 2, an event 4, 6, 8, 10. And this shows how the network is growing in time, isn't it? So we can do this with, for instance, loops and domains, and see how those evolve in time, isn't? So, for instance, Berezovsky in Israel proposed actually in Norway, so proposed some years ago, the idea that there are elementary functional loops. These are little snippets of protein structure that somehow combine with other snippets to form the fold structures. These are the elements generators of domains, ultimately. And this is what you're seeing here. A catalytic residue acts as a foci for helping a functionality of a loop structure. This is a very ancient process, obviously, but it could be recent too, isn't it? And that's something that can be tested. And then this kind of assembles with others to form an active site of the Rubisco catalytic domain, and there you have the fold, the modern fold there. The same you can say with domain organization. You can say, well, I have a domain, and this domain can co-opt another domain, it may be older or younger, and both join together to form a multi-domain protein. And remember, there is a substantial number of multi-domain proteins in all proteins that are known. And this two-domain protein now can co-opt a third one. And then now we have 3 domains, and there is a topological association, a relationship of the different domains that you can take account for in evolution. So as soon as you're doing this, then you are generating networks that are evolving in time. So a network of an elementary functionome at the top, or a network of domain organization, or of an interactome unfolding at the bottom, isn't it? In the bottom 1, structural domains combine to make multi-domain proteins. In the top 1, elementary functional loop structures combine to make the structural domain of proteins. Both have been published, and you can look at those papers and see what they describe. But the big message is that when we measured modularity, and we measure it with different descriptors, bioinformatic descriptors of modularity in networks. In all cases, we've seen modularity increase with time, which is the prediction of the biphasic theory. But we also seen hierarchy increases in time. So it's both hierarchy and modularity, and it's called hierarchical modularity, what is unfolding in time in these networks. So just to very quickly to show you some examples of this, this is a biphasic network of an elementary functionome, where you have at the left the nodes that describe the loops, and at the right, and in the left you have the domains, and domains and loops are organized by time because we have chronologies to describe this, and we see how the dynamics of recruitment of loop motifs by domain structures is first of all an ongoing and highly modular process, meaning that the processes did not stop, did not originate at the beginning of life and then that's it. The process continues. It's permanently generating new modules that are being added to the repertoire. And not only that, modules in the bipartite network behave as integral communities. You don't see the results there, but in the paper you will see the details. And when you're looking at clustering coefficients, there is a trend of network hierarchy and modularity and evolution unfolding in these networks. You can look at the networks as a waterfall diagrams, and I like this diagram because it's like the flow of water follows the flow of time, isn't it? So the network is organized in such a way that you can see now waves appearing, like a wave that is associated with p-loop structures, and a second wave, number 2 there, that is associated with the wing helix. That's the origin of the EFL8, and then Rubisco, which is such a crucial and central enzyme for Earth, appearing later on there, about 1.5 billion years ago. Well, this is a similar situation with multi-domain proteins. We can generate waterfall diagrams of domain organization on a radial diagram. In the middle are very ancient structures, in the outer part of the radial diagram are the recent ones, and here you have how a progression in time from top to bottom of the different network. These are snapshots of the network, but if you want to see the CX network, which is a special type of network out of the many we have that describes the sharing of domains more than the connectivity, you can see how at some point at some point, sorry, I had to jumpstart this. At some point, there is a Big Bang occurring roughly 1.5 billion years ago. This is the rise of multicellularity, the rise of eukaryotes, the global change on Earth of how things organize. Organisms are becoming bigger, and they are abandoning the realm of microbial life. And there you have the Big Bang occurring. And you can see that as a structure already in the network. So this is something we already proposed many years ago. And here's the cover of the journal Structure that reported that Big Bang. But there are many Big Bangs in evolution, Okay, so now you have an understanding of the kind of power that chronology in a network that is evolving has to dissect a number of interesting things about language. So the entire talk is driven towards the understanding organization of language, and seeing where that organization of language is associated with organization that we see in biology. And so in very recently, but I've been kind of hammering this for a long time, we've been putting forth the idea that there are analogies that we can drive between linguistics and molecular biology. Linguists are much more advanced from a computational point of view than biologists, but that's by far. And they already have seen interesting patterns in their language makeup. And biologists have been really slow in taking some of this, though in the 2000s there have been very interesting proposals put forth. So I find a link on the progression of language organization that goes from lexicon, syntax, semantics, and pragmatics. But I can find links between that progression with a progression of module to structure, to function, to fitness. And in order to have structure, you need to have a module, you need to have a unit, that would provide that vocabulary. In the same way as in order to have a syntax, you need to have a lexicon, isn't it? You need to have that elementary elements, making up the whole. And so let's go one by one of these. So the lexicon first, what is its definition? It's the set of signs used to communicate meaning. And I find a lexicon module correspondent. For instance, the morphemes, are the smallest grammatical units of language that combine to form units. So if you go to this database that's called the Cognitarium, you can actually go and explore individual morphemes, like the morpheme morph, which means shape, form, and you can find it in a number of words. It's an allomorph, amorphous, anamorphic, anthropomorphic, and so on, including some that for biologists are very familiar, like polymorphism at the bottom there, so there's the diversity in the shape or form of something, isn't it? So that's polymorphism. That's the smallest grammatical unit of language. Well, we can say, wait a minute, the loop would be the smallest structural unit of a protein structure. Why? Because it's the unit that somehow can combine to form domains, and these domains combine to form higher level structural entities. So this smallest structural unit has so many of the proteins of that morph, of morpheme. EFL8 represents something similar to that morph, isn't it? So let's look at syntax. What is syntax? The systematic combination of the inventory of signs, like the morphemes, according to semantic rules of grammar. So we know about grammar, we've been through school, isn't it? So you know about verbs, nouns, and so on, and how they can be organized by physical precedence there, words combine to form language expressions, dissected with many ways, but one of them is called the catena. The catena is this network. As you can see there's a network where I am dissecting the sentence. Word organization alters meaning, isn't it? So, and that word organization I am kind of ordering by physical precedence. So I'm going from left to right, word, organization, then organizations followed by alters, alters followed by meaning. And then I'm organizing it by dominance. The verb alters somehow organizes the entire sentence structure, So you have a dominance relations on the y-axis, a physical precedence on the x-axis. Well, I can do the same thing with domains in multi-domain proteins. So this is an aminoacylthianate synthetase protein responsible for the genetic code and interacts with transfer RNA there at the left, and the entire purpose of this molecule is to charge specifically an amino acid corresponding to what it sees in the transfer RNA molecule. So the catalytic domain is the central element, and is a historically old element of the entire structure. And then it gets decorated by anticodon binding domains that interact with the transfer RNA, and then by accessory domains, and there's a physical precedence because they are organized in a very defined way, and if you can change that definition, as some other aminoacylthianate synthetases have done, then you have a different organization, but the dominance axis is maintained. The catalytic domain is the central synthetic element of this enzyme. Without it, this is nothing. And then you decorate with specificity and other things. So domains are structural units combined to form protein structures. Very much word combined to form language expressions in language. Let's look at semantics now. What is semantics? Meaning of signs and their combinations at different hierarchical levels of the message. So now it's not so much the makeup of the language structure, but the meaning of it, why that language is there to begin with. And as soon as we are here, that now we have this semantics function correspondence that I'm driving, isn't it? So now the morpheme word, so this is what is called a cognitive synonym, isn't it? It's called a synset. And what you're seeing there on the left is just a synset of morpheme that you can find in a database called WordNet that's in Princeton that somehow dissects the English language. And as you can see, there are all these hyponyms and hypernyms. It's a hierarchical structure, that describes semantic relationships. So you see, morpheme can be an allomorph, or it could be a free morpheme, or it could be just a classifier. It's much higher level understanding of what that morpheme is all about, isn't it? So in the same way, we have gene ontology in biology doing the same kind of semantic function. So it provides lexical and conceptual semantic statements describing biological functions. That's the goal of gene ontology, isn't it? And it has a hierarchical structure. It's a direct acyclic graph, define a molecular function level at biological process or at component level. And you can, given the world of gene ontology terms surrounding a molecule like the aminosyltRNA synthetase, then you can construct a word cloud like the one you have there that describes the catalytic, accessory, and anticodon binding domains with all their functionalities. It's metabolic, it's intracellular, it's associated with an organelle, it requires binding. You're somehow dissecting the meaning of that molecule as soon as you are into this level, And now we go to the highest level, pragmatics. What is pragmatics? Well, it's the context-dependent relation of signs and sign user according to rules that govern successful and unsuccessful communication. Now, all of a sudden, the sign user, the person that is responsible for conveying language, like I am doing now, now that person is using language in a particular way, to draw attention to the receiver end, which is you down there, hearing this talk. And for that, you need a triangle of effective communication, that describes the context of how the individual elements, that I've been describing till now, contribute to meaning. And so one proposal that I made in this, what I call the triangle of effective communication, is this 3 vertices, in this triangle of communication. A vertex of relevance. Am I conveying relevant information? That's things that make sense. I'm persuading you, that what I'm saying makes sense, and am I using the right words, the right elements, the things that are understandable? That's the attraction element of this triangle. In all this, there is all these rhetorical, referential, emotive, cognitive, relational, reflexive components that are all in trade-off relationship. And some they are describing different elements of the kind of conversation I'm having, in the same way, we can think of a molecule as a molecule that is driving through effective molecular communication. Let's say an aminosyltRNA synthase has to catalyze. That means it has to add that amino acid and load it into its molecule. That requires a budget. There's a chemistry reaction there going on, and it has to be policed in some way. It needs to be a gatekeeper. That means that not any amino acid can be charged, isn't it? There's a discrimination component, and it's associated with relevance, isn't it? Catalyst is associated with persuasion. You need to really persuade the meaning of what you're doing, isn't it? And then you need a mechanism. You need to have a molecule in such a conformation that will turn it into a machine, You know, that mechanism is needed to accomplish that effective molecular communication. So as you can see, there's correspondence here that makes a lot of sense. Let me illustrate that very quickly. This is Jacobson's structuralist model of linguistic communication he put forth in the 50s. As you can see, it's a model based on Shannon and Weaver's understanding of message and transfer of communication, this transfer of information. You know, so a sender sends a message, and then there's a receiver. The sender has to have an emotive function, you know, able to communicate and convince the audience. The receiver has to be attentive, conative, you know, need to understand that the message is coming. There needs to be a context, a referential function there, and the message needs to have a rhetorical function that will transfer that information. And at the same time, there needs to be a context and a code. You know, there needs to be an understanding of how the communication unfolds. So this is again the Neo-Grecian inspired triangle, Grice advanced Jacobson structuralist model of language by defining a quantitative principle on the top, you know, which is related to persuasion, a relative principle relation to relevance, and an attraction contact qualitative principle on the left. Well, just to illustrate how this works, this is an example with a slogan from the Australian Transport Accident Commission. You know, so that it's when you are driving in Australia, we'll see this kind of things. If you drink, then drive, you are a bloody idiot. You know, very Australian, you know, message. But look at this. You is the conative, is the receiver, isn't it? And you're stressing with the word, if you drink, then drive, you are a bloody idiot, isn't it? So you're stressing that element of the triangle. Then drink, then drive is the referential element, isn't it? It is in the relevance, the reddish part of the triangle, isn't it? And that's important because it's what really catches your attention, makes it relevant. Oh, it's drinking, it's driving, it's something that I'm interested in. But then look at this, you, the conative, are a bloody idiot. And here you have a conative behavior that causes death. You know, that bloody word is very crucial because it implies that if you drink and then drive, there is a consequence to it. And the consequence is bloody, isn't it? But bloody idiot it's an idiomatic expression that is rhetorical, that it stresses that relevance aspect of the thing. Of course, you need to have an attraction component too, because you need to convey all that message. But the whole goal of this is to stress the relevance, action-driven vertex to impart emotional impact. So that's how language and semantics and pragmatic works. This is the context of the whole thing. And we can think of the same thing with any molecular system. Like for instance, here we have a catalyst that is related to persuasion, a gatekeeper that is related to relevance. Here, discrimination is crucial. The attraction element is the mechanism component. And the example I'm going to use is the ribosome. You know, you need to have peptide bond synthesis occurring, and that needs to be a chemical, faithful mechanism that is related to budget, you know. But at the same time, it has to, not any amino acid should be bound to any other. There needs to be a gatekeeper function. That's this discrimination, the relevance function. And of course, there needs to be a mechanism. There needs to be a way to turn and produce all the different peptide bonds that are necessary to make a protein. So as you can see, there is an element that is so crucial. Now, let me now make bring you to the context, because all of this is about context in language and biology. And here I'm bringing Lewis Carroll with Through the Looking Glass. He was a subversive pragmatism at that time when he wrote Alice in Wonderland. But he brings this subversive centrality of meaning. You know, and this is Humpty Dumpty talking to Alice. You know, when I use a word, Humpty Dumpty said in a rather scornful tone, it means just what I choose it to mean, neither more or less. But then Alice says, but the question is, whether you can make words mean so many things, isn't it? So, well, if you decide what a word is, you know, then there would be innumerable meanings, isn't it? But then Humpty Dumpty responds, the question is, which is to be master? That's all, you know. And this is wonderful because, you know, it just distills the essence of the entire thing, you know, of persistence, you know. In order for you to have a persistent system, whether it's biological, whether it's language, whether it's anything, you need to have that effective communication occurring, isn't it? So this is Sokos naming rules. And for those interested in bioinformatics, you will know that you have to, in order to have addresses in the internet, for instance, you need to follow a protocol that is memorable, that is secure. You know, you can't have anybody entering and modifying that, because that there will not be reproducibility. And if it's decentralized, that provides an aspect of strength into the persistent name. Well, it turns out that conversation has the same idea. And I already described the triangle of persistent communication. I described to you the triangle of molecular biology, but there is a general triangle. And the general triangle is a triangle where the vertices are one of economy on the top, robustness on the right, and flexibility on the left. And you need to have those three elements that are embedded in all triangles that you could build of communication of any kind. And those three elements, economy, flexibility, robustness, are central in that kind of thing. So in 2013, Lyudmila Yefremova, Jay Mittenthal, and myself got to think on trying to understand systems in interaction with environment. After all, we are all surrounded by environment, no? And already Jacob von already there was an idea that that systems were interaction with the environment, and Miller proposed in the 1970s this living system theory, where there would be information flow going through a system, and at the same time a matter-energy flow going through systems. We can understand matter-energy flow as, for instance, metabolism processing all the matter elements and molecules so that we can generate energy, and then spend that energy to produce things valuable for cells. But the same way there's information flow that processes a number of elements needed for that specificity, for instance, that we need. So living system theory does that for you. But we propose that an organism is interacting with the environment, and it's visualizing that as an environment as a scope, you know, and that scope is composed of two elements, an umwelt and a gap. So the umwelt is the set of environmental signals that undergo sensory processing. And the gap is the set of environmental signals that do not undergo sensory processing. If they don't go sensory processing by a cell or an organism, then the organism must have had incorporated those signals that existed into their makeup, so that those signals may not harm the system if we want to have a persistent system. Therefore, we associated the scope with economy, umwelt with flexibility, and gap with robustness. And in doing so, we defined a triangle, and this is the triangle of biological persistence, where we have the vertex that defines the matter-energy budget, that flow in the living system theory of Miller. And then we would have an economy that defines an economy vertex. And then we will have a flexibility side of things defined by umwelt, and a robustness side of things defined by gap. And in doing so, you would have a persistent system living within that realm, you know, of trade-off relationships. And in an analysis using a number of descriptors that were molecular, we came to place individual organisms or groups of organisms into different parts of this triangle. So organisms like archaea and bacteria push boundaries that are related to economy more than the other alternatives, and therefore they're closer to the economy vertex. Organisms like metazoa that generate so much diversity push boundaries within themselves in interaction with environment that are closer to flexibility. And the same goes for, and in contrast, plants, for instance, decide to use smaller repertoires, but they're highly robust, and therefore they're pushing the envelope of robustness. This is one of the examples of how we tested this kind of hypothesis using omic vocabularies. This is using domains and gene ontology terms. And you can see that indeed, the distribution of those terms follow exactly that tendency, you know, from archaea being highly economic in the way how it deals with things and highly robust at the same time, then bacteria somewhere in between, and then incredible flexible structures being produced by eukarya. So okay, so getting close to the end of my talk, but let me now introduce laws of language. They exist, and they are statistical laws, things that are statistical recurrences that occur in language. And what I'm going to claim now is that those statistical laws apply also to biology. Well, you may know the SIP law, the probability distribution. The frequency of a word is inversely proportional to its rank. You may also know the Heaps law. It's called a developmental type law because it involves times, though time is not one of its variables. However, it's the idea that a vocabulary grows as document data grows. So if you have more and more texts of being produced, you know, books and content in the internet or whatever, as this is growing, the vocabulary is growing too, isn't it? So all of a sudden there's a new word appearing called fefe. What is that? Okay, well, it's a new word that appeared recently, and it's now a part of the vocabulary that you find there, isn't it? It's been added to it. And it follows the Heaps law. And then there is a functional type law, the Menserath-Altman law, for instance. This is the idea that the greater the whole, the smaller its constituents. If you have something big, the parts of it are smaller. You know, the bigger the whole is, the smaller its constituent parts. And this was found in music, and it was found in Menserath by utterances, the length of an utterance in speech. Well, it turns out that all these laws are embedded in biology. The SIP law is part of scale-free Sipsian regimes that you will find in the frequency of structural domains occurring in proteomes. This was already found a couple of decades ago. We reported that in 2003. The Heaps law is just a vocabulary of domains, scales with the domain reuse in proteomes. This is something that you can very quickly generate given a domain census in proteomes. And you can see it has different regimes. There is a viral regime that follows the Chinese regimes of words, regime 2, because the Chinese has several regimes, three regimes of the Heaps law. Archaea has an English regime, bacteria has a Chinese regime 3, and eukarya is extreme. You know, just that it's out of context. It's going further than any language we know. So it's here you can see the different regimes, label 1, 2, 3, and 4. We also discovered a Menserath-Altman law in domain combination. The length of structural domains in a multi-domain protein decreases with domain number in superkingdom dependent manner. And there you have an example, Candidatus methanoregula, out of the 60 proteomes that we studied. And we saw interesting patterns coming out of this that in all cases embodied persistent and compression of information. Because after all, the three laws that you see there are expression of compression. You know, here we are looking at scaling regimes. You know, viruses are somehow embodying in the Heaps law a primordial vocabulary that is then repeated over and over in the other languages of bacteria, archaea, and eukarya. But that viral vocabulary encases that commonality, you know, that is embodied in the Heaps law. So So let me visualize this with an example here. Domain accretion in proteins and proteomes we find is linked to persistence and compression. And how do we do this? Well, the Menseroth-Altman law, here we have Gabriel Altman, which was the mathematician in the 60s that turned the intuition of Menseroth into the law. And it's basically a continuous distribution model describing the probabilistic relationship between the size of parts, in this case is Y, and the whole system, the entire system measured as the number of constituent parts, and that will be X. So like for instance, if you have a protein, that's the whole system, you can measure the size of that protein by the number of domains it have. So a larger protein will have, with more domains, will be larger than one that has less domains in general, of course, with variations. And that is going to be part of the landscape, statistical landscape. And that's the equation. This is a general equation. It has two terms, but you can fit the law by focusing on the first part, let's say A times X to the B, or by focusing on the second one. So you can choose, and people have been using the two variations, which describe a hierarchical component, basically, of language. So we applied this to domains in proteins, and you can go to that paper and you're going to see the details. But we re-elaborated that equation and when we made a function of domain accretion, when we proposed a persistent function, which was function of domain accretion K. And it turns out that that persistent function has two terms. And those two terms, one reflects the matter energy cost of adding domains, and the other one reflects information gain in intra and intermolecular interaction. So as you can see, we are trying to somehow use this persistent function that describes the Menseroth-Altman law to fit the triangle of molecular persistence that we were proposing, and see whether that can be described with this mathematical in this mathematical way. And what you have here with this kind of balance there at the bottom, you have this the triangle unfolding, isn't it? As more matter energy budget you add to the system, then more you need to split between flexibility and robustness, and that you can only unfold by unfolding information, isn't it? And it makes sense, because the more domains you have, the more combinatorial landscape you have, the more kind of words you're going to have, and meanings of sentences made out of these domains, and pragmatics out of the domains. So the more matter energy budget you add into the system, the more information you are going to be unfolding. And this then manifests in proteins that have different kind of makeup. And when you look at the archaea proteome and the bacteria and the protista, plant, fungi, and metazoa, and how the different components of the equation of the Menseroth law unfolds in this triangle, it turns out that we have, again, that beautiful progression from archaea being highly compact and metazoa being highly diverse and flexible structure. So we've done something similar with history of terminal domains. And it turns out when you are looking at the history of the gene ontology terms, it turns out that you see unfold again the same patterns. Look at the age of terminal terms on the right and how they appear in evolution, very similar to the protein domains. At the same time, look how in the top graph in the middle, are fusion indices, which are just how many, how domains are distributed in lineages in the world of organisms, how that distribution is very sparse compared to the distribution you would find in eukarya. And then as you, in the three plots in the bottom, as you look at how the time of origin of geoterms unfolds in time, you can see how there are two components, two decreasing contrasting evolutionary modes. One decreasing losing trend that pushes everything down as time progresses to the bottom there, in archaea and bacteria very cleanly. This is fostered by vertical dilution in lineages. And then you have at the same time an expansive trend pushing, these indices that describe dilution upward, fostered by horizontal processes of recruitment. So eukarya are the masters of recruitment and are the masters of combinatorial game, and that's why you have that wonderful placement in the flexibility vertex of the Menserath-Altman, mathematical construct. So, so as soon as you use the gene ontology terms, then you can use different levels of the hierarchy of the gene ontology hierarchy, and to make statements about distribution of those gene ontologies in time. So there you have the trees, and there you have the Venn diagrams describing the distributions. And in all cases, you see something common coming from this. Archaea and bacteria are highly compressed in their vocabularies, the molecular functions, and eukarya is not. So we are seeing this unfold with numbers, but we can use now the pragmatic view, of perhaps Catena to unfold in a word cloud, the terms, the gene ontology terms dissected into this dependency grammar Catena inspired strategy that I'm proposing here. So this is done with level 2 molecular functions of hundreds of proteomes analyzed at the gene ontology level. And you can see that there is a progression in time. This is the time of origin, relative age, coming of the terms. So this is the vocabulary of life unfolding in time from left to right, and a precedence, unfolding from bottom to top, and the precedent connects important words to each other, like binding to iron at the beginning there in the left. That's the primordial element of any kind of vocabulary that you need and language that you need in biology. You need to bind and receive signals, isn't it, in some way through information and matter energy constructs. And you do that with binding, but you start with what? With ions before going into small molecules, heterocyclic compounds, or organic cyclic compounds that come later on. So by studying this very carefully, this graph carefully, you're going to see that the vast majority of central things for the cell are universal. They're all red, isn't it? Common to everything. There are not many that are unique to bacteria and archaea here, but there are many related to eukarya. Those are the ones in gray. All the structural constituents that are necessary for accretion, muscle, myelin sheaths, extracellular matrix, nuclear pores, complex scaffolds of different cases, egg coats, and so on. All these are very derived things that came quite late in evolution that were added to fundamental discoveries from ontological perspective, or for instance, a structural constituent, or a channel, that is something uncovered that is common to bacteria and eukarya. So this is a way to look at the vocabulary of life, dissected with gene ontology terms at the level 2. But you can do this at level 1 or at different levels. So this is my end. So it turns out with RNA evolution, we can do the same thing. We've generated, and we publish all this, chronologies that uncover the origin and evolution of the ribosome. It turns out the ribosome started to unfold 3.3 billion years ago, and it generated the core 1.3 billion years ago, the complete core. But it was actually 3.1 billion years ago where the core achieved all the functionalities of interacting with the small and large subunits, processing the messenger RNA, recognizing the transfer RNA, generating the protein transferase center, and all that was established in that structure that you see here at the right. And that's the first component, isn't it? But then there is a second component. Now the molecule continues to grow, and as it's growing, it has to produce an exit tunnel where the proteins are kind of funneled in, isn't it? And why are they being funneled there? They're being funneled there because we need to generate proteins that are more resilient, that fold more reliably. And now we know that there is co-translational folding occurring in the ribosome. This is research for the past 10 years. And it's all related to the exit tunnel that is being formed, that appeared after that core was generated. So, that allows us to now understand why we see this biphasic pattern that you see lower there on folding time that we find when analyzing the domains of proteins. So it turns out that first, everything is to increase the speed of folding at the beginning. And then once proteins are multi-domain, then folding time increases because, of course, there are longer molecules, so they take longer time. But now we have a fantastic machinery to do that, a complete exit tunnel structure facilitating the folding of the molecules that come out of the ribosome. So this, again, this phase 1 structure and the phase 2 structure that then provides that diversification that is needed for that folding. So it's functional at the end, isn't it? So one thing that we found when we were looking at RNA evolution, you know, and unfolding the chronologies, was that by character state reconstruction, we were seeing a diminishing return principle unfold, you know. It showed that short helical segments tend to become smaller and larger ones tend to become larger. And that was kind of curiosity at the beginning, but then as soon as we started experimenting with Menserath law, we understood that there was a Menserath law embedded in the size of an evolution of the ribosome and other RNA molecules. Look at the transfer RNA. As time of origin of the individual segments of the RNA molecule unfold in time, the size of the segment decreases in time. So as transfer RNA molecules grow in evolution, their stem components become smaller. Well, it turns out that the 5S ribosomal molecules also grow in evolution, their stem components become smaller. And the same goes for the RNase-P RNA molecule fundamental for processing of transfer RNA. As they grow, their stem components become smaller. So definitely that looks like a Menserath law, isn't it? So can we fit that Menserath law to a Menserath-Altman equation? So that is the question. So, well, it turns out that ribosomal RNA does the same thing. As RNA molecules grow in evolution, their stem components become smaller. So we've done that. We saw that there was a strong correlation, both Pearson and Spearman driven, that showed there was this strong association and push towards an economy of scale of all these RNA molecules. Well, what we did is just use the second term, the decay function term of the Menserath law, to fit basically the Menserath law fit the data to the Menserath law, and it shows that the helical stems become smaller when their number increases in molecules. So this is a principle of evolutionary economy or compression that we are really seeing quantitatively unfold in the ribosome, for instance. And so what is wonderful here is that now we can understand language compression. And it goes back to Krakauer in 2002, where he was theorizing the idea of what was compression in biology. He was thinking that compression was economy-driven optimization favored by stable, rapid, and efficient propagation processes of biological information. And indeed, I think all our results are compatible with this view. This is a global view that is definitely embedded in all the biology. So what are the take-home messages before I close here? Hierarchical modularity, and that somehow explains the generation of modules in biology, and of course the generation of modules in language, but also explains why they become more hierarchical and granular, isn't it? We have more of them, more of different kinds, more of them arranged in hierarchical modular manner, and more granular, as time progresses. The second take-home message is that persistence imposes evolutionary constraints of economy, flexibility, and compression. Now you know about economy, you know about flexibility, you know about compression and what it means, but it's all driven by persistence, isn't it? The organism wants to further its existence in time. And by doing that, it has to find mechanisms to do that. And those are the ones that then ultimately unfold, for instance, in archaea, bacteria, and eukarya, and their compression in terms of the vocabulary. And finally, the lexicon, syntax, semantics, and pragmatics of language pervade biology. Yes, I hope I've convinced you that there are a number of analogies and a number of relationships, that link elements in biology with elements in language. And all those elements are really compressed in evolution, both in language and in biology. So providing us like a general theme driven by evolution in terms of, what are the ultimate goals of this evolutionary principle. So with this, I close, and I leave you with this interesting theme from Peter Gabriel, which I think is perfect manifestation of the process of accretion that in his take, which is very interesting. But with that, thank you very much for your attention. And of course, I will welcome all the questions that you may have. So.
Thank you very much, Gustavo, for this wonderful talk. And we have time for some questions. Miguel, you have questions?
Yes, I have. Gustavo, thank you for the inspiring conference. Many students are thanking you for that. I think it was very inspiring the way you think and you apply your thoughts to your science. It was very great. I would like to make to you a challenge. If you look at eukaryotic proteins, you take the domain and ask what is the coverage of the protein that is represented by a domain. I think you will note that sometimes not sometimes, but mostly the eukaryotic proteins, the most recent ones, they have a globular domain, but they also have a big domain conserved that does not resolve, is not structured. It gets structure when it combines with another protein. While it's functioning, while it's associating. On the contrary, for bacterial ones, the structured portion is almost 100% of its length. Mm-hmm. So what would you suggest about that eukaryotic are doing something different when they have those domains that will get the structure only when they are bound to another one?
Mm-hmm. Yeah, you raise quite an important aspect that we need to dissect. We identified domains using hidden Markov models that have been becoming more perfected with time, and of course those models use the fold structures that are known, isn't it? That the genomic revolution and structural genomic revolution managed to generate, isn't it? There are two encouraging aspects of that, let me tell you. One is that we are not finding new folds as our genomic exploration unfolds, and it's massive. Our genomic exploration is becoming massive at this time, but there are not so many new folds arising, So we are stuck there with about 1,200 and something defined at the fourth superfamily level. We may be around 2,000 and something, and those numbers are not changing dramatically, meaning that what there is there is there, and then probably, we come 1,000 years from now, and our research would have advanced incredibly, and perhaps we will fill in all the gaps. But the tendency of that curve is telling us that we are arriving to a plateau, and that plateau is encouraging because it telling us that using our methodology, our hidden Markov models, which are really powerful, allows us to identify those domains in the way how we define them in a particular, in a very exhaustive way. This is even better for loops. In terms of loops, you know, the number is large, but there have been no loops uncovered, you know, after 2014. No, 2014 was the maximum, and that's it. Nothing. All the genomic revolution has not provided extra information with extra kinds of loops that would appear, meaning that we've as bioinformaticians, we must be congratulated because we've done very good job, at least at the loop level. Now, there are many other things we don't know. We don't know how genes are generated de novo. You know, they've been for the last 10 years. We've been studying all that aspect of generation. You know, how little snippets that are actively producing small little peptides at some point coalesce into larger things that produce then proteins. Because we define protein in a very particular way. We use a threshold, and things that are below that threshold we ignore. And all those are probably very active things that are in the genome and that are accounting for the things that you say are in that percentage that is not being covered. That is the problem, I think. It's not so much that our methodological identification of structures is failing. It's more than we don't really understand, you know, why those other entities, possible open reading frames that would be targeted and for generation of structure, are not producing the structure when we run the Hidden-Markov model. So yes, same occurs with viruses. With viruses is even worse, because with viruses we can only target a really low percentage, you know, of all that incredible diversity. And it's really tempting to suggest that we cannot see folds in viruses at that level, because perhaps viruses are the drivers of generations of novelties in our planet. And that's the reason why they have so many in the making there, you know, that at some point they co-opt, you know, into some new fold that then is shared with the cells. And we have evidence to support that that process is actively occurring from viruses to cells. But I think you bring a very important subject matter, because yes, in all studies in genomics, we've been constrained by our tools. The same goes with evolution. We've been constrained by the beauty of how well all these trees can be constructed, you know, and we trust those because they've been with us for decades already, and they're providing good output. I would suggest, given all what we are learning from COVID-19 now, how beautifully all these phylogenetic reconstruction processes are being modeled now, you know, and generating all this information of the different clades and variants of concern, and everything is coming beautifully from the methodologies of phylogenetic reconstruction that are acting at lower levels of structure, because of course they're acting at the sequence level, isn't it, with a population that is changing at incredibly levels, you know, because viruses, RNA viruses are changing at an incredible rate compared to all the rest of life in this planet. So we have the ideal system, and we can put it to the test, because we see what happened in 2020, December, to what is happening mid-2020, when the big variants of concern started to arise, and how things are occurring now. And we can see how, and we can retrace back. We can do character state reconstruction and double check it and see that the methodology is working. So, all this is wonderful, because it's supporting the evolutionary process, it's supporting our phylogenetic methodology, then claiming that what we are doing when we're building chronologies is not completely out of the box. It's something that is going to be producing something reliable. But at the same time, we have so much to learn. And I think you hit on several points of importance, which would be, you know, what is the set of ORFs that are, lacking lacking proteins that we can use to identify them and understand what they're doing. You know that, and our Hindenburg models are very effective in some cases in eukaryotes. You know, like human. You know, 80%, if not more, of proteins are hit by the Hindenburg models, but there's still this 20% is problematic, isn't it? And if you go to some other organisms, you know, basal eukaryotes, for instance, then there you have 40% only, you know, so telling us that there's something there that for some reason, you know, needs to be explained. You know.
Catherine, would you like to ask something?
Questions? Hi, Catherine.
Hi, Catherine.
Are you looking for a question?
No, yeah, I don't know. Yes, I was, yeah.
I just found this completely fascinating, and I'm interested in the connection between the network dynamics that you're describing and attractor dynamics. Is that part of the story that can fit into the semantics and the whole correspondence there between language and biological structure?
I think so. Yes. Because attractor dynamics is basically it's one aspect in systems biology, and you want to see how dynamics can be mapped onto our static view. We have a static view with genomics, isn't it? And we can not only turn it dynamics by doing the kind of things we're doing with COVID-19, isn't it? We are just grabbing over and over sequences, and now we have millions, and we can see how that virus has been changing with time, isn't it? So, but in absence of that, you know, of that level, then we have to use some other contraptions and some generalities. Like we were looking at, for instance, I mentioned the zip flow. Well, the zip flow is ultimately scale-free behavior, isn't it? That you will find very common in the networks in biology. So any network like metabolism has clearly a scale-free component, you know, to its makeup. And I wouldn't be surprised that that scale-free is associated with some attraction dynamics that are occurring, because the networks are not. It's a description of an interaction, isn't it? And we know interactions are ephemeral. You know, the thing occurs now, but it changes to, you know, some minutes afterwards, and so on. In fact, I have not shown, but we've been spending millions of node hours in a big supercomputer we have here in campus, which, by the way, you have there in the slide, Blue Waters. Now it's going to be updated and going to be done even better. But so we spend more than 1.5 million node hours running molecular dynamic simulation of loops. And our entire goal was to turn those loops into networks, because you can do that. You can turn dynamic movement into network structure. And then trying to see whether we can map that into a space of network makeup, how modular it is, how scale-free it is, and how random it is, because it could be random too. You know, networks. There is a randomicity in all this that we are describing. And remarkably, again, the dynamic behavior that was occurring at nanosecond level was expressing hierarchical modularity. So it means that you know, as soon as you, Catherine, are pushing the envelope of attractors and dynamic behavior, that is something hard from a bioinformatic point of view, because it requires a lot of computational resources and right algorithms that we don't have. You know, we've been unfolding those chronologies, and this is a hard problem. And let me tell you why. You are mapping how a network is adding nodes, and not only that, it's adding nodes and connections. You know, and this is the most simplistic of nodes of networks we can envision, because we could also map differential equations to them and other things, and then we will have a collective of all this that we need to somehow sort out in the network structure, unfolding in time. But if we just take a structuralist point of view and we see how the network is changing in time, and that's why we saw that hierarchical modularity, that biphasic element, unfolding in time. Very primitively, we are finding how individual events of growth are tailoring this network structure. That to me is fascinating, because I think that's where the real future action of everything in biology is going to be. We want to see how things are functioning, and they are being embedded into time frames of nanoseconds to billions of years. All of those are playing roles, but just that we naively think it's only the present is the present. The present embeds all the history that goes back in the system, and without that history, we cannot understand that present very well, nor make predictions for the future either. So that's the goal of evolutionary bioinformatics in general, isn't it? So.
Some people have questions in the chat. If they want to open camera or
I can read one.
Live.
Yes, I can read one here that I can see. It says from Elionai Mura. Can I go? Elionai Mura Cordero. So there is any direct correlation between smaller structures and specialization of the function of complexes. Wow, that's also quite an interesting question, especially because I saw that you put that question very early in the talk, even before I unfolded the Menserath-Altman law in the ribosome. So that means that you were kind of predicting some important aspect in the story. But it is true, what are the functions of those smaller structures, for instance, in the ribosome, that are appearing now? What are they doing? Let me tell you this. It took a lot of effort, and Savio knows because he organized a meeting about the ribosome, and there you have all the luminaries talk. But there's a limited number of structures of this incredibly large structures, which are the ribosomes, and this is going to change because we have new methodologies and so on that are coming our way. But the numbers is limited, and so it took a long time to get the bacterial ribosome. Then we have the archaeal ribosome, and then we have several of those. Then we have the Saccharomyces cerevisiae ribosome. They were all rather conserved and of the same size, roughly, But then it turns out that when the human ribosome was obtained, as a structure, it was just a massive thing with all these legs coming out of the ribosome. We understand the ribosome as this very compact structure. Well, the human ribosome changes all that. So why is this? Why are these molecules becoming larger, with evolution? And why are they unfolding all these new tentacles and all these things, coming out of them? My immediate answer would be, well, because all the cell is becoming more structured, and all those tentacles are new novel interactions that need to be established with all that novel complexity that is being generated. So, and for that you need novel things, because the ones that you already have been cooled by evolution, frozen in time, put it in that way, and therefore they cannot go, they cannot be used readily. So you need to add more things, but adding more things in that growth implies spending more energy to build them, so they're costly. And that's where the Menserath-Altman structure comes. The Menserath-Altman tries to push that boundary towards economy. So where economy of scale now becomes an important factor, because it's much easier to synthesize a smaller ribosome that will have the same function, you know, than having a big, gigantic, unstructured element, you know, unfolding there. Now, why would all these little additions be added to the complex? Well, they should be added because obviously there are new interactions that need to be accounted for. That is telling us that we are ill-equipped yet to understand interactions unfolding at the cell level. You know, so that means that in order for us to index the role of each individual novel helical stem that is being added into a ribosome, we don't have appropriate gene ontology terminology labeling their contribution, the novel contribution, because we haven't done the experiments in the bench, I bet. So, so this is a long response to your point here. But it's one that is important because what we are just doing now is unfolding some general mathematical recurrencies that we see over and over again. It took us a long time, for instance, to apply networks to biology. It took us Barabasi and his colleagues to push that envelope, and everybody went into networks. And now we know much more about them. But it's still, applying dynamic behavior modeling to networks it's in its infancy. And so we cannot understand why those networks are unfolding in the way they do. So,
Gustavo, I posted a web tool that we produced here, where we have the time, not the time, the clade of origin of Go terms, the ancestrality of Go terms. You might like to take a look.
Yes, please. Yes.
I don't like time in million of years ago because I'm mostly fan of Einstein and space time, not time. So I understand like frames that are passing by, but sometimes they go fast, and then slowly. And so we express the ancestrality as the clades in image. That you share terms or genes with other species, like if the gene occurs only in man and the chick, and so we call the origin at Amniota clade. If fish is included, Euteleostomi. If Drosophila, we go to Bilateria. You can turn that into millions of years ago, but it's expressed like that. The ancestor clade in human lineage, where the Go terms first occurred. Maybe you could play with your ideas on that thing.
Would you mind, Miguel? Would you mind sending that by email to me? Because the problem I cannot, for some reason, I cannot copy and paste for some reason. I don't know. But
okay.
Yes.
I will send.
Yeah,
including if you would like, you can have the MySQL dump all the database. Okay.
Okay.
So it's a tool for you to see if you like the idea. It has the origin of function, not functions do not exist since ever. Some functions are appearing along the time. But also it has the origin of processes. This is very difficult, has many filters. It's very dangerous to say this process has occurred here. But we tried with many filters to process the origin of Go terms, including the processes, right? We had another study. We picked up the complementary strand and translated, okay? And we sent it for a prediction of alpha helix and beta strands. And our conclusion is that if you originate a gene from a sequence that is not translated, it will have pretty much the same density of alpha helices and beta strands. So the problem is to modulate that structure, but this is not difficult to originate at least the secondary structures. They are there pretty much in the same density that you observe in the real proteins, you know? We did that experiment, and so we think that's very easy to originate a structure. Of course, this structure has to function, but then you need the loops and, you know, the catalytic site, but the structure itself, it's already there, even in the intergenic domains, but mostly in all the areas where you have the same concentration of ATCG, you will have pretty much structures that would fold into a nice 3D structure.
Yes. Yeah, there is a universality in the structural makeup of proteins at the secondary structural level. And we have not published this, but it's in the making. We've seen an immense Altman in the secondary structures in proteins. And so therefore there's definitely a principle of economy going on at that level, you know, and that's probably why you're seeing this unfold at different levels, you know, in the standard structures, but also in the ones that have not been acquired yet. But you can access that information through algorithmic methodology, isn't it? So you access that information, and you still see the pattern arising. So which makes it even more interesting, because that means that this language is, you know, it's embedded in what I was saying. It's very ancient. It's incredibly ancient, but at the same time, it's still unfolding. Meaning that you have novelties, and that's where the exploration of novelty from a theoretical point of view is so important, because there are very few people that have been pushing that envelope. And usually it comes from physicists and not biologists, you know, that have been pushing the envelope of how novelty is being generated, you know, and this goes back to Bass and the Bass equations, and the idea of how you know, different chips are kind of evolving and overtaking the population of computers in the world, and then a new chip appears, and then just you know, and all these are biphasic processes, by the way, you know, where you have adoption of a new novelty, and the adoption at the beginning takes a long time and long effort to achieve, but then the adoption overwhelms the entire population and takes the whole thing over. And so all those kind of mathematical formulations are already there, you know. It's just that they've been applied to physics in general more than biology, and only recently I think people are kind of understanding the need to revisit all that, you know. That's why I placed that slide of Mark Pagel, because he finds exactly that. He finds that models of innovation are embedding language. You know, of course, he's studying language, which is a biological expression, isn't it? It's not language; is human language. So it's very advanced, you know, novelty that came to be in biology. But he makes it explicit there. I wonder whether, you know, we could make this explicit at biochemical levels too, and at molecular evolutionary levels too. It's going to be hard, because there's the mathematics is complex, isn't it? To transfer from language to biology. But anyway, so.
More questions from the audience? I had a student that started with me, then developed everything with Sergio Mattioli, the origin of folds, and we like prion protein. The prion protein fold occurs only in vertebrates. It's not older than the origin of the vertebrates. I don't know if you see something like that, some proteins that are very new. Some folds that are very new.
They're very new, yes, yes, yes. And also intrinsic disorder. We've been studying the intrinsic disorder, and we'll be publishing soon about that. It's also recent. Intrinsic disorder is a highly derived feature that we see in proteins. You could also see from one of the slides. One of the slides shows the location of, unstructured proteins in the CATH database, and which pools all the intrinsic disorder proteins. And where does that set of proteins land in the world of architectures of CATH? In that tree, and it turns out that it lands halfway in evolution. So, meaning that it's a highly derived feature, isn't it? So, very much as what you were saying about proteins that have these behaviors that prion-like or prion-generating structures, so seems to be a novelty that is recent. Now, in all this, we need to be careful too, Miguel, because the problem is that new things co-opt old things. That has been universal that we've seen in metabolism and everything. And I wouldn't be surprised that prion proteins contain motifs that are very, very old,
No, I mean the basic fold, the suprasecondary fold, you cannot see before.
You cannot see, exactly.
You only see in vertebrates. Of course, you see many vertebrates, but you have a common ancestor. But you cannot see in Drosophila.
Yeah.
No way.
Yes. So
I have another challenge for you, because I work with pathways. So when you look at a pathway like interferon response, response, you have many parts of the response that are captured from other pathways that are working in the cytoplasm, etc. But at some points in euteleostomy clade, you have originated the receptor, the proteins that deliver the signal to the nucleus, and the transcription factor, but only for interferon alpha only. Later on, you have more interferons appearing, and they appear even in placentary organisms. They have some interferons that will appear only in placentary organisms. So from fish to placentary organisms, you have the interferon, but you already have the response. So you are correct, they capture things, but I'm impressed that at some point there is this thing, interferon response, and then later on we can see that it's improving. So I'm in love with the appearance of new things. And for pathways, if you work with pathways like KEG, so we have a tool that has the ancestral of all proteins in KEG. I will send it to you too. So I'm fascinated with the origin of a new thing. Do you explore that when a new function appears, when a new thing appears? Well, of course, they are generated from ancient ones, but they change something, that they create something new.
Well, we have not done what you've done, which is go into specific examples, and which is really valuable too. Our views have been always very global. And the reason why they've been global have been because we wanted to see, to try to approximate global repertoires of things, take the entire thing and see what kind of pattern. So we've taken metabolism as an unit, or we've taken protein-protein interactions in domain organization as unit. We've taken the gene ontology database as unit, and so on. But in reality, our global statement of hierarchical modularity being an emergent property explains what you were saying, explains that, at some point there's going to be a module arising, and this module you need to mine with bioinformatic tools in order to identify, because if not, you won't be able to see out of the noise that is there, But once you identify the module, you see that it becomes recurrent. You can see it perhaps transferred to some other thing, and the module does not need to be physical in terms of things associated with each other. Like for instance, pathways in metabolism, they're unlinked physically, isn't it? Though they are multi-enzymatic complexes and so on. Of course, they've been described, but from a chemical point of view, isn't it? It's just it's an open system,
Yeah.
And you have the same thing with gene ontology. It's an open system. The molecules are free to take from wherever. They're not bound to bind their networks to a physical milieu. I would say that is not the case of neurons. No, and as soon as you are studying the brain and so on, then the neurons are bound to a physical milieu that place them into a physical relationship, and that could occur also with molecules like complexes and so on. There is a structural binding of things together, but pathways, as you were saying, they're not. So that means that.
But at some time, a key protein appears that makes all those proteins to execute a function, like immunology, like interferon response. You have very old ones, ancient ones, but at some point, one protein appears that connects in a representation like a pathway that can do something, and this has an origin, a specific origin, the clade of vertebrates,
Yes,
it's determined by a couple of key proteins, and then the pathway starts to function.
Yeah.
Without them, without interferon receptors you don't have the function. But at some point, bang, with 5 proteins and all the other ones, you have a new thing. Mm-hmm. Immunology is good for that.
Yeah, exactly. Well, I think everything is compatible. All your results are really compatible with what I've shown. But it's compatible with recruitment, because
recruitment.
The recruitment has been a pervasive thing that people in bioinformaticians actually have been making explicit. The molecular evolutionists have been really concerned about recruitment because it messes up their nice phylogenies. That's the problem, because it's an horizontal force that kind of borrows from different parts of origin or times of origin interesting things. It brings something that is very old with something that is very new and puts them together and to function into something completely novel, as you were saying. That is the challenge in all this, because every chronology that you generate, like the ones I've been showing to you, the chronology applies to your level of organization and your data set. As soon as you try to apply that kind of chronology to other statements, then there's a risk. And the risk is that there may be things that you are missing, in that complex mapping. And what you're missing is your hierarchy. As soon as hierarchy arises and you have a new level, what happens with that level? How does it map to the level that was there before? and that has been a problem with structure in itself, you know, because you have sequence mapping to structure and how that landscape unfolds. And theoretically, it's a big problem. It has been a big problem, and also limited by our methodology, isn't it? You know, the people use lattice models of structure because they're simple, and then you can computationally track all these things. They're theoretical constructs, but nevertheless, they're telling a story. They're telling a story that the recruitment goes all the way from the basics of molecules all the way to what you were explaining on your pathways, isn't it? Just there's some point the things are ripe for things to combine and form something novel that then unfolds aerobic metabolism, like the ones I showed there, you know, or unfold behaviors and functionalities in vertebrate lineages and so on. That it's just an expression of or revealing, by doing this kind of analysis, revealing novel hierarchical levels that we need now to sort out, you know. And you know how difficult it is to sort out, for instance, a domain in a protein. You know, there've been a lot of effort trying to do that, and dozens of domain classifications put forth that continue to arise and so on, because we want to perfect the definition of the module. But that's a very basic module. But what about the modules at higher levels? There is no systematic analysis of many of those levels that I know of. There have been attempts I've seen of you know, higher structure levels of things. You know, I remember, for instance, Bochel and Teichmann in UK putting forth the idea that we should categorize kinds of cells. Those are modules too, isn't it? How many modules of cells do they exist? You know, how can we, you know, bioinformatically identify them? You know, that's an interesting, important question, isn't it?
You know, Gustavo, we will have a seminar in January, I think, from the series. If you put BRAZWEBINARs, that's the title of the series, you see the program. And there is a scientist that will show a modeling of the full cell, all the proteins, and then all packed together, and the cell interacting is a new thing. Is from the author of Vin Autodoc from California. And I think it will be interesting because he will show the 3D structures of all proteins interacting in a cell. It will be,
which is amazing.
Nice
amazing feat.
You're inviting all your students. If you wanna bring people to our seminars, you are welcome. Okay, bring your students, people from your university.
Thank you very much.
The same place we advertised your seminar, we have other ones. Savio, you are in the dark.
I just want to say, Gustavo, thank you very much for this amazing talk. And thank you everyone for coming. Miguel, do you want to say something about the next seminar?
We only have seminars in January. If you place BRAZWEBINARs in Google, you'll check them. Okay. So it was great. I think it was very inspiring for our students, Gustavo. Thank you for coming, right?
Thank you for the invitation. It's very kind of you. So definitely. So
yeah, so let's close this session. Gustavo, I hope see you soon. I see you in February, right?
Yes, of course. Yes, we'll be in touch for that that thing too. So yes, and so let me see how I do.
So let's close the session, Miguel.
Yes. Okay. Thanks, Gustavo.
Thank you.
Thanks,
Thank you.
everyone.
Yes, let me stop to share. Thank you very much. Okay,
Bye.
and keep safe because we need that. We need to be careful with all this that is happening. So, and it's been a pleasure seeing you and discussing all these interesting subject matters. So,
Yeah.
okay, see you later. Bye-bye.
Bye-bye. See you guys. See you in January for another international seminar. Check I think there is a seminar on Nagai, a very expert on health and genome. Is a big expert on health and genome. Come on January for that. Podem chegar em janeiro para esse seminário aí, que o cara de Stanford é brabo, né, em janeiro. Eu não me lembro quem que fala no final de janeiro também, mas já tem conferência em janeiro, fevereiro e março, muito brabas mesmo. O cara do AlphaFold vai vir aí, viu, gente? O cara do AlphaFold fala no final de fevereiro e vai falar de resolver estrutura a partir da estrutura primária, né? Inteligência artificial aplicada a folding, acho que é no final de fevereiro. Então, tem uns seminários brabos por aí, podem usar como das suas instituições, viu? Pode marcar como evento de vocês, porque aquela data está fixa, pode promover aí quem quiser. Então, Teletubbies, hora de dar tchau. Vamos lá. Tiago já desligou aí a gravação, né, Tiago?
Oi, professor, ainda gravando. Vou encerrar aqui, tá bom?
Ah, tá bom. Achei que quando deu um flash aí, a gravação encerrou. Beleza. E qualquer coisa, a galera sabe que a gravação fica no canal da Bioinformática UFMG, né? Bioinfo UFMG no YouTube.
Exatamente.
Né? Beleza. Tiagão, obrigado pela ajuda de sempre aí, importantíssima. Eu fiquei sem luz aqui durante um tempo, só na bateria do computador e o no break no modem da net, cara, funcionou legal pra caramba. Né? Recomendo. Claro que pode ser que eu esteja enganado, mas assim, recomendo um no breakzinho no modem nesses tempos de chuvarada. Você está longe da Pampulha, né, Tiago?
Exato. Vou encerrar aqui, tá bom, professor?
Tá bom, obrigado. Tchau, 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 gente.
Tchau, pessoal. Até semana que vem.